Thank you so much for the kind introduction. Hello, everybody. So yes, today I would like to talk about calcium channels and store-operated calcium entry. See, yes, you see the presentation. So yeah, calcium channels, store-operated calcium entry, and more particularly in the context of endothelial cells activation by pro-inflammatory cytokines, most particularly IL-1 beta. So this project has been done in Dr. Trebak's lab, hosted in the Vascular Medicine Institute of the University of Pittsburgh. So I don't know if you are familiar with store-operated calcium entry, but I would like to go back to a quick background of how this pathway works, because this is the most important calcium signaling pathway in non-excitable cells. And as today we are talking about endothelial cells, we can expect this store-operated calcium entry to have a fundamental function in this cell type. So physiologically, store-operated calcium entry is induced by several agonists, typically VEGF, thrombin, carbachol, ATP, acetylcholine, that will bind to receptors and activate the phospholipase C pathways that will lead to deacylglycerol and IP3 production. IP3 will bind to its receptor at the membrane of the ER, and that will lead to a massive calcium depletion from the stores. And this decrease in calcium from the ER will be perceived by calcium sensors of the ER, STIM1 and STIM2, that when calcium is low, this protein will activate it, clusterize, and propagate to the near the plasma membrane, where they will interact with different partners, and notably calcium channel ORI. The binding of STIM on ORI channel will be responsible for the activation of ORI channels that will lead to this massive calcium entry that will change the behavior of the cells. And we will talk about the functions of ORI channels and store-operated calcium entry in endothelial cells in a couple of slides. But now I would like to show you the actual protocol that we use in the lab and that you will see during the whole presentation to measure SOC in the different manners that we have to investigate it. So here we are looking at Fura2 recordings. So we use this calcium dye that is cytosolic to evaluate the modification of calcium fluxes over time. And the first part of the protocol is done without calcium in the extracellular compartment. That allows us to verify the calcium release that is necessary to induce the pathway. So here, when we applied thrombin, you can see this nice calcium release from the ER that then goes back to baseline. And when we reintroduce calcium, we have this massive entry that is coming from all the channels of the plasma membrane, all the calcium channels that are open at the plasma membrane, but most particularly, we expect OI channels that to be the main driver of this calcium entry. So to be certain that the signal we are measuring is due to SOC and not some other channels that are open, we use gadolinium at 5 micromolar to block OI and be sure of what we are measuring. At the end of the recording, we use ionomycin just as a loading control to be sure that the cover slips are properly loading and we have the right dynamic range in the recordings. But with this kind of measurement, we have kind of an indirect information of how ORI channel is activated. So we also do patch clamp recordings using the whole cell configuration. This time, we do not use agonists to trigger the whole pathway. Instead of this, we use BAPTA in the pipette in very high concentration, here 20 millimolar of BAPTA. And that allows chelation of the available calcium and will be responsible for calcium depletion from the stores, which will therefore activate store-operated calcium entry and the opening of OI channels, which is what you can see here. After 100 milliseconds, the current starts to develop and then gets this nice steady state. So if we do the subtraction between the activated state of the current and the baseline, we can highlight this nice inward rectifying current that is selective to calcium, and that is the ORI-induced current, which is called ICRAC. The recordings I'm showing you right now is done in in an overexpressed system. Unfortunately, in native condition, crack is very small. We can expect it to be around 1 picoampere per picofarad when it's big in native conditions. So we need to use a trick to enhance the size of the current if we want to measure it, let's say, again, in native conditions. So to do so, we use this DVF protocol. So how does that work? ORI channels are also permeable for sodium. And so by using sodium and the properties of sodium, we can enhance the actual current that we are recording. So we applied steps with a solution that contains mostly sodium instead of calcium. And we can see here during these steps that are highlighted here in red, the development of the current. That is just here this spike that activates very fastly and then inactivates. And by doing the subtraction again, like before, we can highlight this nice inward rectifying current that is this time a sodium-induced ICRAC current. So I told you that there is different partners to mediate SOC. We need steam, 1 or 2, and we need also ORI channels. In reality, there is, in mammals, 3 ORI channels, ORI1, ORI2, and ORI3. ORI1 has 2 different isoforms, ORI1 alpha and ORI1 beta. And for the cell type we are talking about today, which is the endothelial cells, previous work from the lab and from other teams showed that ORI1 and STIM1 are the major carrier for store-operated calcium entry. So we will just focus on these 2 partners in this talk. So ORI1 and STIM1 will be our focus today. Actually, there is some work that have been done in endothelial cells and try to understand what is the role of store-operated calcium entry in this cell type. And basically, there is 3 main functions that have been identified over the 2 last decades. The first one that is very important is the control of nitric oxide production by store-operated calcium entry. In this study, the authors created a STIM1 knockout cell, endothelial cell line, which lack NO. And as a consequence, the vessels in the mice, when they did the same experiments in the mice model, they saw an increase in contractility and lack of relaxation in the blood vessels. The second main function related to store-operated calcium entry in endothelial cells is the ability to promote angiogenesis. Typically, SOC will be responsible in all the steps that are associated with angiogenesis, meaning migration of the endothelial cells, the tubulogenesis, and also cell proliferation. And the last main function associated with store-operated calcium entry in this cell type is the regulation of leukocytes, monocyte infiltration, and also cell permeability. So typically here in this graph, you've got the vessel permeability, endothelial cells permeability in wild type or in SI control or in SI STIM1 cells. And as you can see, this spike here is associated with the permeability and the recovering of this increase in endothelial barrier permeability. STIM1 cells, STIM1 knockdown cells exhibit less permeability than the controls. So definitely there is this idea that maybe store-operated calcium entry could be involved in the regulation of how cells can pass through the endothelial cells and so cross the vessels. And this is corroborated by other works showing that under inflammatory context. Here, showed with LPS, lipopolysaccharide, which mimic bacterial infection. SOC promotes the recruitment of leukocytes and monocytes on the endothelial cells. So because of all this diversity of functions within these endothelial cells, we can expect that a change, an increase or decrease in SOC, can lead to a modification in the behavior of endothelial cells dramatically and leading to disorders. The two main functions here, the two main, let's say, misbehaviors will be called endothelial activation and endothelial dysfunction. So for the definition of it, let's say that endothelial dysfunction is a lack of the ability of the vessel to make the contractility and the relaxation, which is related to the NO regulation part of the store-operated calcium entry we have seen before. And the endothelial activation is more related to the ability of endothelial cells to promote the expression of adhesion molecule, typically ICAM, VCAM, E-selectin, which will be responsible for the recruitment of leukocytes and monocytes on their surface. And this endothelial activation is the one we will be interested in today. It's promoted by inflammatory molecules and could be sustained by excessive oxidative stress. Why is it interesting? Because this endothelial activation is necessary for the initiation and then the progression of vascular disorders, especially atherosclerosis. Atherosclerosis, that is one of the leading causes of death worldwide, that I've seen its prevalence doubled among the last 2 decades, most probably due to our way of living, the sedentarity, the way we are eating now, and eventually the stress. So we will focus more particularly on how chronic inflammation, here illustrated with IL-1 beta, is able to promote endothelial activation. Why IL-1 beta? For 3 reasons. Because the concentration in IL-1 beta increases significantly in patients with vascular disorders, because the concentration of IL-1 beta is directly correlated with the progression of atherosclerosis, and because it has been shown in clinical trials that using antibodies against IL-1 beta receptors can reduce dramatically the recurrence of cardiovascular events. So this IL-1 beta pathway seems to be very important in the progression and the fate of the patients that have this kind of disorders. But unfortunately, there is no data actually available about the role of SOC in IL-1 beta induced endothelial activation. So this is the question we were asking for this project. Does SOC participate in IL-1 beta induced endothelial activation? So the first thing was to evaluate endothelial activation. So to do so, we've got this very easy protocol. We took endothelial cells, seeded the cells in a 96-well plate, and wait until the cells reach confluence. So we've got a nice monolayer of endothelial cells. The next day, the cells are treated with IL-1 beta or different drugs that we will talk about during this presentation. After the 24-hour treatment, we expect the endothelial activation to be present. The cells are washed with fresh media, so they are now without any drug remaining. And we put Jurkat T cells, leukocyte, on this monolayer of endothelial cells for 2 hours in the incubator to allow the leukocytes to bind on the endothelial cells. We wash 3 times to remove all the weakest adherent leukocytes. And then the count helps us to understand how activated are the cells. So first, what we did is to check that indeed IL-1 beta was able to activate endothelial cells. So first, we looked at leukocyte adhesion on the endothelial monolayer. And as you can see, there is a significant increase in leukocyte adhesion in the cells that were treated for 24 hours with IL-1 beta. And this was correlated with a significant increase in ICAM and E-selectin protein levels in the cells. Then we measured SOC to evaluate the potential involvement, modification of SOC during IL-1 beta treatment. And what we observed was a significant decrease in store-operated calcium entry after IL-1 beta treatment in the endothelial cells. To confirm these results, we performed patch clamp experiments using the DVF protocol I talked about before. And as you can see, the current is significantly decreased in the IL-1 beta treated cells, confirming the previous results. Because the treatment we performed with IL-1 beta is 24-hour long, we thought that maybe IL-1 beta was modifying the expression of OI1 and STIM1. So we checked the mRNA and protein levels for OI and STIM. And surprisingly, what we observed was no difference in the mRNA and the protein levels for both OI and STIM. That leads us to the idea that IL-1 beta was decreasing the activity of STIM and OI. And we thought about three main hypotheses to explain this. The first one was that maybe OI could be oxidized by reactive oxygen species, leading to a decrease in SOC. Or eventually, the phosphorylation status of OI and STIM is changed because of IL-1 beta pathway activation. The last possibility, and nonetheless, was eventually that the puncta formation of STIM proteins was affected, or that OI was not available at the plasma membrane because of its internalization. But among all these hypotheses, we prefer to focus first on the OI1 oxidation hypothesis. Why? For two main reasons. The first one is that ROS is a hallmark of endothelial cell activation, and it's known that chronic ROS exposure can decrease significantly ROS in endothelial cells. So we could expect ROS to be involved in the pathway here. So how do we challenge this hypothesis? We measured SOC in IL-1 beta treated cells, and we exposed the cells to TEMPO, a non-selective ROS scavenger. And what you can see here is that TEMPO is sufficient to rescue completely SOC in IL-1 beta treated cells, meaning that IL-1 beta promotes ROS is responsible for a significant decrease in SOC in this system. And interestingly, when we looked at leukocyte accumulation on endothelial cells, when cells were treated with TEMPO under IL-1 beta treatment, we saw that TEMPO was able to abolish the induction of endothelial activation by IL-1 beta. So we thought maybe the decrease in SOC was associated with the IL-1 beta pathway and leading to endothelial activation. To evaluate this hypothesis, we created two knockout cell lines, an OI1 knockout cell line and a STIM1 knockout cell line, in which OI and STIM were respectively significantly decreased, and where we saw a significant decrease in store-operated calcium entry. And interestingly, what we observed when we evaluated the endothelial activation in these cell lines, we saw a significant increase in leukocyte adhesion in the OI1 and in the STIM1 knockout cell lines. And the amplitude of increase was similar as the one from IL-1 beta. And we didn't see any additive effect when we treated the knockout cells with IL-1 beta, meaning that most probably the decrease in SOC is part of IL-1 beta pathway and leading to this endothelial activation. To confirm that the activity of the channel is involved in this process, we use pharmacology. To do so, we use SANTA66 and GSK 7975, which are two OI inhibitors. And these two OI inhibitors were able to recapitulate the effects we saw with the knockout cells, which means an increase in leukocyte accumulation in the endothelial cells in a range similar to IL-1 beta, and without any additive effects when we did the co-treatment with the two drugs. So that means that the decrease in SOC is definitely part of IL-1 beta induced leukocyte adhesion and endothelial activation pathway. Just to be sure of this, we tried to modulate to an increase store-operated calcium entry. And to do so, we used two SOC enhancers, SIPA, which is a SKCA channel activator. SKCA channels are potassium channels, and if we maintain their activity, we will increase the driving force for calcium and then enhance calcium entry and SOC. And AI65 is a OI1 enhancer that binds to the channel and maintains it in an open state. So we use these two drugs, and what we observed is that these two drugs are able to abolish the effect of IL-1 beta on leukocyte adhesion to endothelial cells, confirming our previous results. And this mechanism is not exclusively present in the cell line we studied until now. So all the results I showed you were done in human coronary artery endothelial cells. And we've got similar trend in pulmonary arterial endothelial cells, meaning that both systemic and pulmonary endothelial cells behave the same when they are treated with IL-1 beta. But also, the mechanism is true in airway smooth muscle cells. So this seems to be more consensual than just in endothelial cells. So to this point, this is the pathway that we are describing, and we would like to know how we can go from a calcium signal to the change in the behavior of the cells. So the canonical pathway related to SOC is SOC, calcineurin, and fat. So we thought that maybe if SOC is decreased, then calcineurin activity will be decreased too. So we checked for it. We used two calcineurin inhibitors, cyclosporin A and FK506, and the results we obtained were similar to the one from SOC inhibition, which means that calcineurin inhibition is able to increase leukocyte adhesion to endothelial cells and seems to be part of IL-1 beta pathway. So as I said before, after calcineurin, the next so the next partner should be NFAT. But because we are describing here a decrease in the activity of the pathway, we can expect that NFAT will not be involved in the process we are describing right now. And as you can see here, we are looking at NFAT nuclear translocation induced by tartegargin that is used for inducing SOC. And we used control cells, IL-1 beta treated cells in red, and then all the drugs that we described before. Because of the pattern, you can see with all the inhibitors that completely blocks NFAT nuclear translocation, we can discard the idea that NFAT is part of the mechanism in this pathway. So we need to find something else to explain the mechanism that we are seeing here. And because the mechanism we are describing is under a treatment of 24 hours and that we are exposed to ROS production and oxidation, so not necessarily genomic reprogramming here, we thought that maybe it could be wise to check if only a 2 hours treatment was able to modify the behavior of the cells. But as you can see here, 2 hours is not sufficient to modify leukocyte adhesion when we block SOC. So definitely we need something longer that will probably need a transcription factor to promote this, this pathway. So because we are in chronic inflammation situation, we thought that maybe we need to look at NF-κB. Why? Because NF-κB is a key partner of endothelial activation and leads to pro-inflammatory cytokine production and ROS production. So there is also this nice study from 2020 that shows that calcineurin inhibition can activate NF-κB. So first, we tried to look at NF-κB activation and see if NF-κB was involved in this process. NF-κB could be activated in two different ways, in a canonical activation pathway and in an alternative activation pathway. So we blocked both of them to see if NF-κB was involved in the IL-1 beta process. And as you can see here, TPCA1 that blocks the canonical activation pathway is able to suppress IL-1 beta-induced leukocyte adhesion on endothelial cells, while amlexanox that blocks the alternative pathway is not able to promote this phenotype. Also, to confirm the involvement of NF-κB in this pathway, we used SI-AKB alpha, which produce a constitutively active NF-κB. And under these circumstances, we can see a significant increase in leukocyte adhesion on endothelial cells, confirming that NF-κB is part of this mechanism. And if you remember at the beginning of this presentation, I showed you that IL-1 beta was able to promote adhesion molecules, protein induction, ICAM and E-selectin, and we checked if NF-κB was responsible for this de novo production of these two adhesion molecules. And as you can see here, the inhibition of the canonical activation pathway of NF-κB is sufficient to block the induction of ICAM1 and E-selectin, meaning that most probably the mechanism that we are describing is the following. IL-1 beta promotes ROS production that leads to a decrease in SOC, a decrease in calcineurin, which will maybe promote NF-κB nuclear translocation, a genomic reprogramming that leads to adhesion molecule expression, and then endothelial activation and leukocyte recruitment on endothelial cells. So the last thing we need to check to be sure that this pathway is true is that SOC inhibition and calcineurin inhibition are both able to promote NF-κB nuclear translocation. So we did some IF on NF-κB, and we evaluated the nuclear localization of NF-κB under the different conditions that we have seen before. And as you can see here, IL-1 beta is able to promote NF-κB nuclear translocation, as well as SANTA66 and cyclosporin A, both respectively SOC inhibitor and calcineurin inhibitor. And interestingly, TEMPO, which is the ROS scavenger, is able to block IL-1 beta-induced NF-κB nuclear translocation, as well as TPCA1 that we use as a positive control that blocks NF-κB translocation. Just to be sure that SOC is upstream NF-κB in this pathway, we did one last experiment with a leukocyte adhesion where we triggered the pathway with SANTA66 and we tried to block it with TPCA1. And as you can see, TPCA1 can block SANTA-induced leukocyte adhesion on endothelial cells, confirming that SOC is prior to NF-κB in the pathway. So to answer the question that we were asking at the beginning of this talk, does SOC participate in IL-1 beta-induced endothelial dysfunction? The answer is yes, but in a way that was kind of unexpected because of the results that were present in the literature, where usually it's more an increase in SOC that is related to a disease model. Here, you can see that a decrease in SOC is responsible for the induction of endothelial activation and probably of the maintenance of a pro-inflammatory environment that leads to the disease. All right, so I would like to thank you for your attention. I would like to thank all the people of the TREBAC lab, all the collaborators. Unfortunately, they are not all present on this picture, but yeah, I would like to thank them. Also, yeah, I've got this nice picture of Pittsburgh. I don't know if you guys know Pittsburgh, but it's worth visiting. And I would like to thank also you guys to allow me to do this talk today, and of course NIH that funded this this project. Thank you.
Thank you very much. Olivier. The floor is now open for questions, and we already have a couple questions, so I will start with them. But please, I didn't mention at the beginning, for anyone who has any questions, please feel free to use the Q&A tab to write down your questions. So we have a question from Rohit Kumar. Thanks for the nice talk. Curious if Uri channel currents correlate with IP3 on the ER. I mean, probably I'm assuming IP3 receptor mediated calcium release. Are there any plans for follow-up in this direction if this hasn't been done?
So we checked the expression of the IP3 receptors only at the transcriptional level. But what was interesting is that we saw a decrease in IP3 R1 and 2 and an increase in the third one. Also, what was interesting is that not in the HCACs, but in the HPACs, so in the pulmonary one, we had a significant decrease in the calcium release induced by sapsegargine, and also when we use ATP to trigger the release. We didn't check with sepia, but that's something we could do, definitely.
Sounds great. The second question is from Julius Ashut-Kuzak. Method question. Calcium reintroduction protocol is for comparing calcium entry in absence and presence of sapsegargine, yet you simultaneously increase calcium from 2 to 10 millimolar. In the earlier slides, you showed this in your experiment. What was the reason for this?
We use 10 instead of 2 because we want to have the biggest dynamic range to see the effect. When I did the experiment the first time, SOC was very small, and so this is why we choose to increase the calcium concentration outside, and then we just work like that. The cell seems to handle it pretty well, so yeah, we keep it that way.
That's great. Next question from John Lederer. What kind of endothelial cells are you using? Have you tried using capillary endothelial cells, for example?
So it's primary human, pulmonary and coronary arterial endothelial cells that we are using.
Do you know if from previous work by you or someone else in the lab? I mean, do you know if the behavior in other venous as well as capillary endothelial cells is similar or different?
Yeah. It seems to be very similar. Like all the results that I have shown here, we can recapitulate it in UVEC also.
Okay, cool. Anything about capillary endothelial cells? Because this is the particular question I think of Dr. Lederer. So, do you know anything different?
No, we are thinking about doing, because all the results I showed you right now is done on monolayers, right? So we have a collaboration right now where we will try to do 3D models to see if in a flux situation we can have the same outcome. So we hope it will be the case.
Okay, well, we're waiting for the answers. Scott Zavia is asking, does the culturing endothelial cells that do not appear to be 100% confluent in the presence of a SOC inhibitor, Synta66 or GSK compound, result in store depletion over time?
That's a good question. So let me see. Is this question written? Because
it is written now under open, and the rest of the question: how much calcium is present, and is it possible that this induces a misfolded protein response, which could be part of the cellular inflammation cascade?
Maybe, but because we have the knockouts, I think we are pretty safe on the involvement of Orion steam in the mechanism. Yeah, that's what I would say.
Yeah. I mean, this is me just thinking out loud. I mean, would the ionomycin kind of be a way for you to know whether kind of there is a kind of different extent of store depletion?
Maybe, yeah.
Yeah, no, that's a good question. Anonymous attendee is asking, does IL-beta treatment influence Uri channel inactivation? And if this contributes to a decrease that you may see in your system?
That's a very good question. When I did the patch clamp recordings, I haven't seen, but I don't know if I could see it that way. I haven't seen any inactivation, but I was using a BAPTA in the pipette instead of EGTA. So maybe that could be worth trying to redo the experiment with EGTA to see the CDI and see if there is a difference in the 1 beta treated cells.
Yeah, no, that's great. Another question, did you try, thank you for a nice presentation. Did you try IL-1 beta treatment at different time points or only after 24 hours?
So that's also something we would like to do because I think that would be a nice control to see over time the decrease in SOC happening. Because who knows, maybe we just need 6-10 hours to start to see a decrease instead of 24 hours. And also it would be interesting to see with 48 and 72 hours what happens. Because here I'm going a little bit outside of the presentation, but we've got, we have seen that at 24 hours, we have a decrease in cell migration in IL-1 beta treated cells, but at 48 and 72, migration increases. So who knows what happens if we keep the treatment for forever, you know?
Yeah, that's interesting. And I think I can't remember which slide exactly, but you had one of your slides when published by someone else, when I kind of beta treatment, when beta treatment increases ROS. I think it increased, I mean, after the first few hours, and then it sort of seemed as if it may have, it's coming back to where it was starting.
Let me see. It's I can give you the slide. Where is it? Yeah,
I think
yeah,
it was
yeah.
when
The maximum is at 1 hour.
Yeah. So I think it goes along the same ways, whether kind of the what are the time kind of line of these responses might be very interesting. Next question, couple questions from Dr. Guy Salama. What are the beneficial actions of IL-1 beta in its interactions with endothelial cell activation?
Beneficial. I mean, here, of course, I look at it in the prism of vascular disorders, but also it's a physiological response because in the case of infection and things like this, IL-1 beta will help to recruit the human system and then help for um, fighting for this infection. So we can also see it in that way.
Sure. Another question, is there any evidence of a calcium magnesium ATPase on the ER membrane like the SR, but maybe weaker version?
Calcium. That I'm not sure. I don't know.
Sure. Next question from Sergey Balchevsky. Thank you for the nice talk. Did you pay enough attention to check the binding status of numerous regulatory proteins like stem, crack, etc., which can modulate ORI-stem interaction and finally ORI activity?
No, we haven't. This is actually a part that we could and should maybe investigate more because for the moment, the only information we have is ROS decreased SOC. We know that it's the actual actors of SOC that are modulated because when we do the patch clamp, we see a decrease in the current. So it's not a regulation of the driving force because some potassium channels or chloride channels are affected, right? Because this is leveled by the solution we used. But who knows, maybe it's the partners that regulates the interaction that are affected.
Yeah. Question from Damien Bell. With these distant cellular membranes, the plasma and the ER membrane, is there any data on additional cytoskeletal proteins that add to the modulatory mix of these complexes? Would your inflammatory factors have any effect on such sort of cytoskeletal accessory proteins, making it less or more likely basically to happen?
I don't know if IL-1 beta is able to change the distance between the ER and the plasma membrane, but what is sure is that IL-1 beta makes giant cells. When we treat the cells, they are almost 2 or 3 times bigger than the controls. So there's definitely change in the cytoskeleton. They are very they are spreading. They are very flat, spreading when they are the IL-1 beta treatment. So for sure, the cytoskeleton is changed somehow.
Do you know under these circumstances whether the distance between the membranes, the ER and the plasma is changes? No, you don't know?
That I don't know. No.
Sure. Another question from Guy Salama. Have you tested an sulfhydryl reducing agent like DTT, for example, as a protective approach to endothelial cell activation?
No, I haven't.
Okay.
I haven't.
Yeah. Sure. Question from Aida Ashrafzada. Thanks for the nice presentation. Wanted to ask, when you used SIPA, was the aim to increase intracellular calcium this way or enhancing SOC itself?
So the thing is, so SIPA activates SK2 and SK3, and SK2 and SK3 has been described to be related, to be closely, sometimes maybe physically interacting with ORI. That has been done in cancer cells. So the idea is that there is this local potassium channel, this local potassium efflux that will increase locally the driving force for calcium and enhance the calcium entry. Because this is localized in a nanodomain, we expect it, it's in lipid raft, to be more precise, it's in lipid raft. So because this occurs in this particular area, we can expect it's, maybe I'm going too far by saying it's selective to SOC, but the way it's described usually is that when we use SIPA or all the SK activators, it enhances SOC. So we can potentiate this activity like that.
Yeah, I think the rest of the question is also providing some context. Ida says, cause I'm going to do some rescue assays, increasing intracellular calcium with and without treatment in cancer cell lines to study crack role. And I wanted to see if using SIPA can be a better choice, or using, for example, low-level ionomycin to increase intracellular calcium levels. If you have any thoughts on this.
If there is a possibility, I would say you can do both. So,
yeah, okay. The more the merrier, I guess. Last question that we have now from Rohit Kumar. Sorry for the basic question. With co-expression of STEM, did you notice any shift in RI reverse potentials?
Co-expression of STEM, I don't think so. No, I don't think so.
It's still kind of the same standard calcium reverse potential.
Yeah. No, I don't think so.
Okay. I think these are all the questions that we have, unless some of the panelists have any questions. I think we would like to, again, thank you again for really nice talk and very engaging extensive list of questions. I think there is a question. Oh, no, that's just a thank you again for nice talk. So thank you again, Olivier. Really enjoyed your talk and wishing you all the best with your future endeavors. Thank you. And thanks everyone for attending.
Have a good day.